if I may, I'll now move over to David Robertson from Glasgow. I was actually, David, just stalking you on the internet. I didn't actually realize you went to UAB at a roughly same time. I was at the University of Alabama at Birmingham with Shaw Whelan and John Barr as well. I think we might have actually
That's
been
a long time ago, though. Don't let's not talk
introduced to
about
each
this.
other. So, with a proper now introduction for David, I think David is absolute one of the thought leaders in the world for viral evolution and viral bioinformatics. And if you think you've had a bioinformatic idea around viruses, David has probably already thought of it and published it. At least that's what we found many years before. So it's an absolute pleasure to get him on the seminar today. And over to you, David. And thank you for coming along.
Okay. So what I was going to do is talk a little bit about the past evolution of SARS-CoV-2, some context, and then I guess where we're at briefly, and then just talk a bit about our use of machine learning of late. And so these are using these very powerful protein language models. And so in the same way that ChatGPT and these models are being applied to human language, there's a lovely analogy to amino acid sequences, and we've been having quite a lot of fun in that area. So we don't need to spend much time on this, but obviously SARS-CoV-2 evolved the end of 2019, entered the human population. That I think really clear that this happened around the Wuhan market. There's a lot of mis and disinformation in this area, but the reality is we just don't usually know this much about viral outbreaks. Usually inferences into the past, whereas here in real time, connections were made to this market. Initially, that was quite hard because not all of those early pneumonia cases could be connected. But what's really compelling is the home addresses of people not epidemiologically linked to the market also live around the market. So really, in terms of understanding an outbreak, the evidence doesn't get much stronger. And then on the viral genome side, we have extensive information as a result of the first SARS virus. And so it's very easy to link this virus, SARS-CoV-2, into that radiation of bat viruses. And I think what's difficult here is the extent of recombination. And what's happening is over the hundreds of thousands of years that these viruses have been evolving in bats, it's essentially recombining all the time. Interestingly, the signal can be disentangled. So that does sort of speak to some structure genomically to the recombination. So the existence of hotspots, essentially. So you can pull out regions that are what we'd call minimally recombogenic, so they don't have too many breakpoints within them. And so that's not perfect, but in terms of being able to take a large alignment and then pull out sub-trees, that seems to be working very well, and that can be done quite nicely. And when you do that and you start to sort of disentangle the closest relatives of SARS-CoV-2 in the bats, depending on which genome region of the virus you're looking at, you get different closest ancestors. And so that just tells us that the virus that's spilled over, was some recombinant of bat viruses, and there's nothing really circulating that's ever been detected that's close to that across the whole genome. And so again, there's a lot of disinformation and misinformation here. Viruses like RATG13 get focused on a lot, but it's not a particularly close virus. It has a lot of distance from SARS-CoV-2, and there are many viruses now that are very close. And that's exactly what we'd expect. We have a radiation of this virus across China and into Asia. And then another thing that I guess gets missed out when people talk about this in the public domain is it's not a human adapted virus. This virus is an unusual specialist. It's a high degree of promiscuity in terms of the host can infect. And we just happen to be one of those hosts. And there's this lovely resource, SARS-ANI. I don't know if anybody's come across it, but what they've done is very carefully curated the number of animals that SARS-CoV-2 has gone into, and it's astounding. We're not used to viruses emerging like this and being so transmissible to other species, and that we think, and the current hypothesis about that is what's happened with SARS-CoV-2 is it's a specialist for a genus of bat viruses. So these horseshoe viruses that infects are themselves very divergent. And so that switch to ACE2 usage in those bats is allowing it to be successful in many humans. And so all of this really just fits into this very kind of expected narrative of host species spillover. And so you look at this classic paper from Colin Parrish, where he looks at all of the sort of the different requirements for a successful spillover. SARS-CoV-2 really ticks all of these. And the only one that's, I guess, more surprising was this adaptation at the end. But as we've already covered, SARS-CoV-2 was a generalist virus. So that adaptation was initially not really necessary. SARS-CoV-2 is able to get into many different animal hosts. And so that leads on very nicely into sort of what's been happening in the human population. And obviously since then, late 2019, 2020, SARS-CoV-2 has evolved markedly in the human population. And our understanding of this is incredibly detailed because of this extensive genome sequencing effort. There's been a lot of data generated, unprecedented, in fact. We're approaching 17 million genome sequences in the GISAID database. Interestingly, not much happened in 2020. So SARS-CoV-2 is a new human population. There's not much immunity, so the virus didn't change much. There was some changes, D614G. We had an interesting mutation, antigenic change in Scotland, and for 39K. Now this would be considered a really weak mutation, but it did demonstrate the malleability of the ACE2 spike. And so when you place these mutations into the RBD and spike, the bit that binds to ACE2, it was very clear that there was going to be a lot of evolution that could occur. And then, of course, late 2020, we had the alpha variant B117 emerging. And so on the left here, you can see the step change in the number of mutations that were observed. And ultimately, this led to Omicron and to an even more evolved lineage. And so I think the important thing here is there's extensive evolution occurring in the human population. So once SARS-CoV-2 had circulated around the globe, infected lots of people, there's then intense selection pressure to escape host immunity. And when you look at the different variants that have emerged that really have been of significance, there's a quite interesting geographical pattern. So you can see in different parts of the world, so gamma here, alpha in the UK, is quite geographically dispersed, which kind of supports that hypothesis of host immunity changing the environment the virus is circulating, and that's what resulted in selection pressure for very antigenically different viruses. The important thing really is this is an ongoing process, and so most of these past variants are extinct. And what you have then is this replacement of dominant lineages, some of that related to functional change, so Omicron very significantly changing to use the upper respiratory tract and becoming more transmissible. And interesting, that's not that phenotype isn't tied. The virus, different Omicron lineages have recapitulated some of the functional evolution that occurred in other variants. And so you have this ongoing plasticity of SARS-CoV-2 evolution as it both explores functional and antigenic space. And so really what's become clear, and I think there's very compelling evidence there, particularly from wastewater, that the important process here and this sort of normal evolution process for this virus is this evolution in chronically infected individuals. And so people that have long-term infections can generate large numbers of mutations, and that sort of that essentially generates capacity that can then evolve in the human population. Surprisingly, we don't really know how important communities of immunocompromised people are, whether these novel variants are evolving in single individuals or hundreds of individuals, thousands of individuals is still unknown. But probably, some mix of that. And so really going forward, sequencing is now at a much lower rate. In the past, we're detecting novel variants really through growth rates. And so once you see a novel variant spreading, if you're sequencing at a high enough rate, you can detect that. You can detect the numbers. You can use the phylogeny to make an estimate about the growth rates. But now that sequencing rates are lower, often focused around hospitalization, there's a lag in detection. Then just by the time you detect anything at any significant numbers, it's too late. It's going to be spreading too widely. And so we're being really interested in the question, can we, based on a variant alone, use computational methods to infer something about functional properties? And so what we've been using is these protein language models. And so these are these very elegant models where in human language, a token, the sort of the unit usually that you're working at is like a word. in amino acid sequence space, the token is an amino acid. And so in the same way that you could understand, or the model captures something about the local context of a word, and that can be used to sort of understand meaning. There's no real understanding here, but there's contextual links among words that make sense to us. So these models can be applied to amino acid sequences. And so the way that an amino acid sequence context, and here, just remember, we're just talking about letters. So, these are already kind of quite distant representations of the reality of what an amino acid sequence is, but it still works. You learn something about that local context, and that context is really about protein structure. And so the sequence can be represented in what's called an embedding, and that embedding is capturing something about the relationships of amino acid sequences that enable them to fold protein structures. And so these are extremely powerful models. And so here, just using this method from Brian He, called evolutionary velocity, you can study the sort of the links between individual sequences in this embedding space, just to do a reality check that is this representation of a sequence in this higher dimensional space meaningful. And as you can see here, there's a phylogenetic tree on the bottom here, and these new maps of this clustering space. I think absolutely yes, we see a strong correlation between the variants and how they link to the different variants of concern. And so this sort of reassures us there's something sensible going on in terms of this embedding space. And then can we apply this then as a metric to assess functional evolution? And so the clever thing here is, because these methods are premised on natural language, just as with natural language, you have grammaticality, the sort of structure of language, and semantic properties, so meaning in language. It's easy to assess that in a model. If you change a word in the human language, car, van in a sentence, it has very little semantic impact. And so the same thing with amino acids. Some amino acids will have more impact on meaning in terms of protein structure. And so we can use that to assess, going back, the different variants of concern. And so again, here's semantic score in this plot on the left is increasing then with each of the novel variants of concern. And so that again tells us that these metrics are doing something useful. They are capturing something about sort of, I guess, mutation evolution that has that's been then contextualized in this structure function relationship. Interestingly, when we compare the metrics to things like accessibility, so where an amino acid is in a protein structure, so the more accessible on the surface, entropy, so sequence diversity, individual sites, other substitution models, and protein stability captured by delta delta G, the language model metric is not correlated extremely strongly with any of those measures. And so that does suggest that these protein language model metrics are capturing something novel about amino acids that isn't captured by traditional metrics. And so I think that's important. This is a novel metric that we can use to assess function. And so something that's just, I guess, this is Kieran Lam has been playing with this in his PhD. And so clearly using the Wuhan 1 Wuhan market reference strain at this point isn't very useful. And so Kieran's been looking at, can we do a better job of making more dynamic comparisons of novel variants as they emerge. And so the idea is that ultimately is to include this in some of the variant tracking that we're involved in. And so this is just this idea of dynamic reference embedding. And so rather than comparing a novel variant to everything from the beginning of the pandemic, you're just comparing your variant to what's circulating now, trying to get some sense about semantically. So in terms of a functional distance, is this variant likely to be different to what circulated before? It's surprisingly hard to do because you also detect variants that have high divergence and high functional scores, but never took off. And so there's an epidemiological side to this, where variants that potentially could have been important just really never took off because of presumably epidemiological reasons, just wherever they found themselves in the world. Or they emerged at that time when something else was circulating that had a higher fitness advantage, and it's the one that swept to success. And so what's really interesting about these models, and I think the big jump in terms of their utility, is we can apply them to single sequences. And so what we're looking at on the left is essentially in silico deep mutational scanning. So that plot B is looking at the input or the an assessment of the impact of all possible mutations at each position in spike. And so traditionally, when you're trying to assess where variation would accumulate, you need multiple sequence alignments. You need lots of sequences. And so you need to be detecting many different viruses circulating. Because the language model essentially it's been processed or it's processed all existing amino acid sequences, it now has some understanding of evolutionary and structural constraint. Based on an individual sequence, you can use this kind of in silico DMS to get some sense of where variation would accumulate. And so this is showing on the left here, SARS-CoV-2 spike S1, and the scores tend to be much lower than in S2. And that fits with our understanding of these different components of SARS-CoV-2 spike. And so the evolutionary constraints in one is much stronger. You tend to see less variation, and that's reflected by entropy here in S1 versus S2. And so it does suggest that the model is capturing something very useful. What's important to appreciate though, and so this is sorry a bit unclear, but so this is again the same plot here in the bottom with the circles here are the mutations or the variants of concern at mutations at various sites. And so what you get, what others have called this sort of plausible evolution. So many possible mutations are deleterious. They would be disruptive to the protein structure. Many are entirely mild. They would be essentially neutral mutations that wouldn't, the amino acid change has very little structural, very little importance in the structural context. And so what you have then is this sort of band of higher, stronger mutations that are functionally significant. And that's what makes these metrics quite difficult to use in any kind of blanket way. It's not a simple matter of just saying, oh, that sequence has a lot of differences, and so it must be biologically important. It could just be a sequencing artifact. And then really interestingly, we can also we can start to think about evolutionary trajectories. So what we're looking at here is essentially individual mutations, so individual DMS in the model. And what we can do is also then ask the question at an individual site about an individual mutation. Are there other mutations in the protein structure that if they change would make this change more or less likely? And so this becomes very, very powerful. Something that's very well understood is the constraints from the protein structure, essentially epistatic interactions, either direct because amino acid residues are in proximity to each other, or due to protein stability, so for the molecule to maintain some overstability in order to accommodate large changes that are, and we can start to do these kinds of walks in this space. And so this becomes very interesting. And so just some examples here. So E484A is a well-known antigenic change in SARS-CoV-2's RBD region. And so when you mutate that change, it impacts the probability of other changes around it. But those effects are quite localized. And that's very different to the D614G mutation. And when you mutate this site, the probability of other sites that it influences are much more distributed in the protein structure. And so I think this is where these models are starting to become very powerful. You can start to in silico explore evolutionary trajectories. And what this is, and these are well understood processes. It's like, that not every site is as free to accommodate change as other sites, So while underlying mutations may be mostly stochastic with obviously biases from certain types of changes, the accommodation of that change is related to the constraint from protein structure. And as you accommodate changes, that changes your evolutionary trajectory. And so we're kind of starting to explore this now as a way to take a starting point and understand how SARS-CoV-2 its potential evolutionary trajectory. And the kicker for SARS-CoV-2 always is that it's evolving in immunocompromised people. And so we have this really almost or certainly cryptic evolution going on, such that the dominant variants tend to be displaced by some divergent variant, almost a reset from some earlier time point that that is kind of unprecedented in I think, viral evolution at this scale. And so I guess to go over some major conclusions then that we've learned from the pandemic, and the biggest one is we really need to be careful with our assumptions. We were very cut out by SARS-CoV-2 because of its mild, the high rate of mild infections and asymptomatic transmission, and that caused problems in China. It caused problems internationally, and by the time decisions had been made to respond, it was too late. In Europe, SARS-CoV-2 had got to Italy, it got to our ski resorts, and by the time we realized that, it was too late. And so that's something that was surprising because we're used to new spillover viruses being more readily identifiable, being more symptomatic. And so something, that's a major lesson. And unfortunately, this has driven a lot of the mis and disinformation. And so the reason that the Wuhan market isn't linked to all of the early pneumonia cases, and only maybe half of them, is because there were so many intervening asymptomatic or mild infections, something that is well understood at this point. There's nothing hard to understand about that. But to this day, this drives this massive amounts of disinformation. And then classically, we expect a virus that spills over to adapt to its new host to transmit very readily. SARS-CoV-2 clearly was a generalist, and so it was able to spread fairly efficiently in lots of species, of which we just happen to be one of them. And what's happened since 2020 is this incredible amount of virus evolution. So more virus evolution has occurred in the human population now than has occurred over hundreds of thousands of years in bats. And so we're still in a fairly unstable relationship with this new virus. It's still exploring new functional properties. It seems to have a high degree of plasticity. And so in the way that SARS-CoV-2, each variant displaced one another, and each of those had subtly different phenotypic properties. SARS-CoV-2 did the same. Lots of the SARS-CoV-2 variants explored subtly different functional properties. And I think at this point, it's not clear if that will settle down or if that capacity related to the ability to evolve cryptically and for a long time in individuals, immunocompromised individuals or communities, and generate the variation that permits these unusual kind of trajectories to evolve, these jumps, as it were. It's unclear if that will ever settle down. And the clear need then is to control those immunocompromised infections better. And that's got a lot to do with sort of broader health issues. The protein language models are extremely powerful. I think we're just starting to really begin with the sort of, I guess, potential of these. But what is really clear is that the model understands a lot about essentially the feature space a primary sequence has been put into in that embedding. It's capturing lots of things about the relationships of amino acids and protein structure. And that allows us then in silico to start doing things like the deep mutational scanning to explore evolutionary trajectories. And when we talk about prediction here, it's very hard to predict virus evolution. It's inherently unpredictable. But what we can start to do is predict, I guess, the null process in evolution. So given protein structure, given the sort of the need to fold a protein, the model captures what are more probable sequences in that context. And so this is very akin to what's in the way these models have been used for protein design. You can essentially walk sequences to new structural folds. With viruses, actually, we often have a beginning and end point. So we can generate different evolutionary trajectories. And so see, of the trajectories we've actually seen, right, was that, how many possibilities weren't explored? And so this becomes very interesting in terms of understanding future trajectories and the possibility of future evolution. And I guess I think the takeaway is the models are powerful, but they're not as yet at the point where we can really predict functional change fully. But what they will be able to do in the near term is prioritize mutations and variants for experimentation. And I think that's often what people want in the lab. If you start to look at possible evolutionary trajectories and the numbers of possible changes that can be accepted in a sequence, it starts to become astronomical. So I think, yeah, the future of the language model is looking very powerful. This particular work and the language model work is part of Kieran Lamb's PhD, and we have a very nice collaborator in the School of Computer Science here in Glasgow, Kay Young, and that's, you know, having a card-carrying artificial intelligence expert really helps us to go into these new areas. And I think, and if we think about bioinformatics and computational biology, virology, we're very much adopting machine learning and artificial intelligence methods. And this is really the next frontier in these kinds of models as we get better at representing the link between genotype and phenotype, sequence, structure, function in the computer model. So, thanks for your attention.
Thank you, David, for a real tour de force. If there's any questions, please put your hands up, and we'll go to you. David, just a thought. Do you think if you add an antiviral like molnupiravir or ribavirin, it might help you explore that possible sequence space?
Well, I think that's almost like, yeah, the next thing is actually, currently this is sequence to structure, and structure obviously is all about function, and that's mediated through protein interactions. And so thinking about protein structure in terms of normal viral host interactions, but then also once you add a drug, that's another selection pressure. So absolutely, and we've started to do this. It's much harder because you're now trying to retrain the model in the context of an interaction, and so the training data is more sparse. But preliminary data looks like it kind of works. And the intuition here is, you know, the current use of the models is at the level of words. But what we're just doing is looking at the relationship between sentences. And so from natural language processing, it's well understood how to link words together. And so we've applied this next sentence, and Dan Liu in my group has been applying next sentence prediction to those kind of areas. And this is where, and this is the bit that it starts to become, you know, this is the holy grail, is be able to predict interactions and be able to predict drugs. And I think this is happening already for other diseases. And virology, we can catch up. We can use a lot of those models. You know, these are generative models, and these are quite different because you could generate new drugs, you know, essentially is the...
Yeah. No, it just sounds so cool. Right, I'm going to hand over to Jonathan for next question, and then Alan.
Oh, thank, really interesting. So you presented this on the level of the amino acids naturally. I had a question about the nucleotides. So presumably from your analysis of all these different sequences, you may have come up with site-independent frequencies for substitutions, for transitions, for transversions. And I wondered whether you'd looked to see what the difference between the site-independent values are and for individual positions along the genome as far as the potential frequency for transitions, transversions, substitutions.
So the problem with these models is they're really expensive to train. And so we're using Meta's model, ESM. And actually Meta have now, they stopped the project and it's been taken over by a private company, and absolutely, I think what we're looking into is, can we now train RNA/DNA models? And so in the same way that you can, essentially the amino acid level here could become a nucleotide level. And at that point, yeah, absolutely, you could start to look at the sort of substitution potentials. And I think it's a good intuition about what the model is doing, is it in the way that we're kind of comfortable with substitution matrices. It's just a very complicated substitution matrix that's kind of protein structure aware. And to have a nucleotide level model that's RNA structure aware, plus protein structure aware, plus interaction aware. I mean, this is a thing in 5 years, that's where we'll hopefully be, at these, you know, very sophisticated multi-layered models that will understand, you know, it's still machine learning, but, you know, be able to link mutations at different levels and into some context. And I think that's the bit we're really excited getting into. But it becomes difficult because you need extreme GPU capacity. You need, you know, a whole different level of expertise to do that.
Thank you for that question, Jonathan. Last question of the day to Alan.
Yeah, thanks. I don't know if you heard my talk. One thing we did with language models, not me, but colleagues, was we took an existing off-the-shelf model which was trained on words. So ours is text, right, not amino acids. Trained on PubMed and Wikipedia, and we took that and then we adapted it. We retrained it using our text, which is written by small animal vets, which is different. And that increased the accuracy, essentially, of the predictions it was making. And I wonder whether there's an opportunity to do that here, take the existing protein language model, maybe you've done it already, and then train it on COVID.
Yeah, well, I think absolutely. I mean, that essentially that fine-tuning then on specific data sets is really important. The thing you have to be watchful of is just overfitting to, say, SARS-CoV-2 data. But we have a project where we're looking at glycoproteins more generally, and so and I don't think we haven't definitive results yet. It does seem to change the predictions, but for this model, these models, we're just using the generic one so far. But absolutely, I think fine-tuning is the trick, and essentially that's what we're doing in the protein interaction space. You're just fine-tuning the existing model and just saying, look, you're not just the model's not just about your sequence. You actually connect to something, and so the model should start to hopefully learn the relationship between amino acid residues in different sequences, and so that ability to fine tune it becomes very, very powerful. Yeah, absolutely. Yeah.
Thanks.
So thank you both for some really fascinating and utterly enchanting talks today. I just want to end by just reminding everyone that we've got our annual general meeting on the 17th to 18th of September at the Roslin Institute up near Edinburgh. So if anyone wants to come along, please follow the link that Lisa has put in the chat. So thanks to both speakers today, and questions and for joining. Bye all. Thank you.
Great. Thanks for the invite. Cheers.